So I was asked to talk about the work we've been doing to develop treatments for CJD and other Prion diseases, which obviously is of paramount interest to people in this room. Firstly, I need, as we do these days, to make a financial disclosure in that we have an academic, so-called academic spin-out company, which is co-owned by my various employers, as well as some of the founder scientists, and that this company looks after intellectual property that arises from our work. And the antibody I'm going to talk about was licensed to the Medical Research Council by DGEM. So let me start off with, I know you've had some introductory talks yesterday, but let me just go through the very, very basic principles of biology here. All organisms that we know of have genetic material, DNA or RNA genes. This is the information that's passed down from both of our parents. And what these genes do is instruct cells how to make proteins. We're composed of proteins of many sorts. These determine the structure of our body. They are the enzymes which work the chemical reactions in the body. We're made of proteins. And these proteins you can think of as a long string of building blocks, like beads on a piece of string. Each bead is called an amino acid, of which there are about 20 different types. And so the genes instruct cells how to assemble these long beads, these strings of beads. And these, once they're formed, fold into a three-dimensional shape. And it's that three-dimensional shape that gives the protein its unique function. And if something goes wrong with that shape, then the protein doesn't work anymore, and indeed there can be other problems, as I'll come on to talk about. And of course there are many different proteins. In humans there are about 100,000 different proteins that make us what we are, and of course each cell contains many tens of thousands of these individual proteins, and of course as an organism we're comprised of many different types of cell. So that's the basic flow of information in biology, from DNA to RNA to protein, and how those proteins are involved in making the cells in the body work. And I'm introducing that because prions, as we know, are rather different from this. All other infectious agents we know, bacteria or viruses, the germs that cause infectious disease in us, have their own genes, just like we have our own genes. However, prions are simply composed of protein. They have no genetic material of their own. They're made by the host individual. They're made by one of our own genes. And this is one of the reasons why the principal reason why we have no natural defenses against these diseases. If you're infected with a flu virus, for example, your body will recognize this as being containing foreign proteins, and it will make substances called antibodies, and indeed other things, which will eventually, hopefully, cure you of the infection. But because the prion protein is one of our own proteins, our immune system doesn't recognize it as being foreign, and so the immune system is rather ineffective at dealing with prion infections. This also explains why our own genetic makeup strongly affects how susceptible we are to developing these diseases, and also why they're rather resistant to normal means of sterilization, which by and large work on inactivating nucleic acids. So you'll understand, of course, that these diseases affect a wide range of animal species as well as humans. Scrapie was the first of these diseases. It's a common disease of sheep and goats around the world. I'm sure many of you in the United States will be aware of chronic wasting disease, which is becoming more and more of a concern worldwide, as well as in the United States. It arrived in Europe recently. And increasing concerns that this might also become a human pathogen, just as BSE or mad cow disease did tragically in the UK, as I'm sure you all know. So these diseases are also called spongiform encephalopathies, and that's because the brain, when you look at it down the microscope, and this is a rather severe example of that, is thought to resemble a sponge because of these fluid-filled spaces in the brain. But more important in understanding these diseases are these deposits of abnormal protein you can see here. And this is predominantly comprised of abnormal forms of the prion protein, which is a normal brain constituent. That's meant to rotate. Can someone click on the slide? And I do fear that we're probably not going to get the full benefit from this presentation because there are some difficulties with the transfer to the system here. Anyway, don't worry about that. It's meant to rotate and show you its three-dimensional shape. It doesn't seem to be doing that. But this is what the normal prion protein looks like. At one end of it, it's got a part that's roughly, it may not look like it there, but it's roughly spherical. And then there's a long tail, which is not folded into a particular conformation. But during the disease, what happens, and I'm now, as a massive simplification, simply showing the normal protein as a sphere here, and the rogue or disease-associated proteins as these squares. And what happens is that you have a seed, by which I mean it's an aggregate of multiple misfolded prion proteins, and that has the capacity to recruit more of the normal prion proteins to the ends. And so these grow in length as more and more of the normal proteins end up in this misfolded form. These fibers get longer and longer. As they get longer, they tend to fragment, they break in two, and then of course you have 2 seeds. And as they grow, they break, and then you have 4 seeds, and then 8 seeds and 16 seeds. So this is, if you like, a chain reaction, and it's this process that then leads to damage to brain cells and the disease process. Now, it's slightly more complicated than that in that there isn't simply one abnormal way in which the prion protein can misfold. There are multiple different ways, and these are associated, so I'm showing these as different symbols, different shapes here, and these are represented as different strains of prions which cause different patterns of disease in humans. But it's all the same process. Now, a further complication, which is relevant to treatment, which is why I mention this at this stage, is that prions are not all, if you take an isolate of prions from someone who's infected with it, they're not all absolutely identical. You have a predominant species, which is shown by these red squares here, but you also have minor species present. So it's to some extent a mixture of materials. And this is important when you come on to talk about treatments, because there's a problem here that you may develop drug resistance if your drug only targets particular parts of this mixture. Now, I'll just talk a little bit about what we know about the structure of prions. And this is work that really stemmed from work Charles Wiseman did when he was working in my unit some years ago, developing a cell-based assay for prions. Previously, to measure whether there are prions in any material, you had to inject it into animals and see how long it took them to get sick. It's very slow, very expensive, and also very inaccurate. But these cell-based, and we now have this fully automated at the Prion unit in London, so we can do very large numbers of assays very accurately. And because of this, we started again in terms of purifying prions from human brain. We put aside everything that we thought we knew about this and started from scratch. And John Wadsworth in the laboratory developed a completely new method of obtaining full-length intact prions from mammalian brain. And this has enabled us to go on now working with Helen Sable at Birkbeck College in London using electron microscopy to get pictures of what prions really look like. And they, interestingly, are not single fibers, but actually double fibers. This video isn't working either. I don't know whether anyone at the back can click on something. So you see here 2 fibers which are intertwined around each other. So it's rather a complicated structure. In our experience, having a single fiber isn't sufficient. So it's not as simple as just a single fiber growing. It's actually a more complicated double fiber structure that's required to actually get infectivity. And this may be because the single fibers are too prone to being broken down by the normal defense mechanisms of a cell, whether these more complicated structures have presumably have rather more resistance to normal clearance mechanisms and can propagate to cause disease. So, as I'm sure you're all aware, there are 3 different ways you can get these seeds in your brain. Oh dear, doesn't look good. We seem to have lost everything altogether now. Okay. So there are 3 different ways you can get these seeds in your brain. That's the general scheme again. So the majority of people that get CJD have normal prion genes. They've inherited normal prion genes from their parents, and this is called sporadic CJD. This occurs at random in the population, apparently. About 1 in a million of the population all over the world every year will develop this. Your lifetime risk, therefore, is much higher than that, perhaps as high as 1 in 5,000, but it's still a rare disease. You then have inherited forms of the disease, where you've inherited a faulty Prion gene from one of your parents, and this predisposes you to form these seeds spontaneously. Or you can have the acquired forms of Prion disease, where a seed has been introduced by ingesting seeds, as in the case of BSE, or by accidental injection during medical procedures. Now, coming on to talk about treatments, as you're all aware, a number of clinical trials have been done around the world before with a number of drugs. These have largely all been published now, and none of them have shown any significant measurable effects, unfortunately, in these diseases. So at the Prion unit in London, we were tasked at our inception at the end of the 1990s to develop treatments for these diseases. And we have 2 parallel programs. I don't have time today to talk about the drugs we're developing, but I am going to talk predominantly about the antibody drugs we're developing. But before I do that, how would we measure if a drug actually works? In the UK, we've had for some years now the National Prion Monitoring Cohort Study, and so we've attempted to recruit almost all patients developing all forms of Prion disease in the UK, and I must say the recruitment rate has been extremely high. In some years, in fact, it has been 100% of patients participating. This slide's rather out of date. We recruited more than 700 patients to this in the course of the study. Why are we doing this? We're doing this to get an excellent set of so-called natural history data, that is to say how the disease normally progresses in individuals in the absence of a treatment. Because we're well aware from our own experience in doing the PRN100 clinical trial in London some years ago that patients and their families are understandably unwilling to be randomized to receive a placebo and would rather receive the drug. And so that makes it more difficult scientifically to tell whether a drug's working. But if we have an excellent database of how the disease normally behaves in individuals, we can then compare what happens when you give the drug. So this is why we've put an enormous amount of effort into getting these data. And in the process of that, we've also been able to develop better means of monitoring the progress of the disease. And we recently published the MRC Prion disease rating scale, which enables us to get a much better measure of how the disease is actually progressing. I won't go into all the details of this. It's available on our website if you want to see it. It's been translated into multiple languages, and I'm pleased to see it's been widely used around the world. It's very easy to do this as well, because you can actually do it over the telephone, and our nurses do it over the telephone. So that, because our patients are all over the UK, it makes it much easier to do frequent measurements of the scale. And these are showing, each one of these lines is the trajectory of an individual patient as they inevitably, I'm afraid, decline. And the MRC scale, which starts off at 20 in a normal individual, declines to the point of death. And you can see that some are declining much more rapidly than others. And the ones that are declining much more slowly are individuals who have 2 different copies of the Prion protein. And I think most of you will have heard about this, that we have 2 different forms of the Prion protein in humans, the M and the V form. And if you're lucky enough to have inherited an M and a V from your different parents, and about half of you in this room will be MV individuals, the disease, if you get it at all, progresses much more slowly. And Simon Mead in the unit developed a mathematical model based on this. And you can see here that there are 3 different genetic types. You can be MM, VV, or MV. And if you're MM, the disease progresses generally quite rapidly. These are VVs, and these are the MVs. Now, the reason this is important is when you're doing a clinical trial, if you divide up the patients into these 3 groups, you have a much better idea of what their normal rate of decline should be, and therefore when you give them the drug, whether it's having an effect or not. So this enables us to do much more powerful clinical trials than we could otherwise have done. Now, I've already mentioned that we have these robotic systems. We have 2 of these systems in the unit now that enable us to measure Prions very accurately. And there's a lot of effort at the moment going into optimizing these assays for human Prions. And the reason we want to do that is we can then measure prions themselves in tissues and body fluids of patients, blood and CSF, of patients undergoing treatment. So we can tell directly whether the drug is working against the infectious agent itself. This is referred to, the technical term for this is a therapeutic biomarker. And if you have a therapeutic biomarker, it makes your trial much more powerful because you can actually tell whether the drug's doing anything rather than simply having to wait and see whether the patient's clinical features are deteriorating. So what's the best target for a new drug, and how do we tell whether that's a good target? The prion replication, as you've heard from a number of speakers, involves the conversion of normal prion protein, which is a normal constituent of our brains, into this rogue form. What we didn't know to begin with was whether damage to brain cells is caused by a new toxic function of this rogue material, or whether it's loss of the normal function of the protein. And of course, those are not mutually exclusive. It could be a mixture of the two. And, but what we do know is if you get rid of normal prion protein in a mouse, they're completely resistant to the disease because obviously prions can't propagate without, the normal prion protein is the fuel that enables the engine to run, as it were, of prion propagation, and without it, it simply cannot happen. So this was obviously a very good target for a therapeutic. The question is, would it do any damage? So this is a rather technically complex experiment, but the principles of it are really very simple. So although we knew that mice that lacked the prion protein from the earliest stages of life are okay, we needed to know what happened if you stopped the prion protein working in an adult brain to be sure this wouldn't cause damage. So Giovanna Malucci, when she was my graduate student working in the laboratory, prepared two types of mice. She took mice in which the prion gene had been inactivated and then put the prion gene back in again. But when the gene that she put back in was vulnerable to being cut out, that's what these yellow triangles referred to. These are particular sites at which this gene can be excised. And she then made a second set of mice which make an enzyme called Cre recombinase. And this enzyme finds these two sites and cuts out the gene between them. So when you breed these two animals together and produce these so-called double transgenic mice, it goes through normal development in the presence of the prion protein. But when it's at about 12 weeks of age, which is an adult mouse, this gene is switched on and the prion protein is cut out. So they suddenly stop making the prion protein. And the results were reassuring, and that is that these mice were essentially perfectly healthy. There were some very subtle abnormalities in them, but they didn't have any brain cell loss. So we knew we could target the prion protein in an adult brain without doing any significant harm. So the next experiment was to take baby mice, 1 week of age, inject them directly into the brain with prions, and then allow the disease to develop in their brains, then see what happened when the prion protein expression was switched off. And again, that was as good a result as we could have hoped for. This is a so-called survival curve. So you start off with 100% of the animals alive here, and they live normally for a period, and then they begin to die. These are two different lines of mice which make the normal prion protein, and as you can see, they all die. They die at different times because they make different levels of the prion protein, and the more you make, the quicker the disease progresses. But when you look at the double transgenic mice, which have the knockout of the prion protein, these both survive to old age, so they're completely prevented from developing the disease. And if you look at the pathology in their brains, and now you look at the mouse brain down the microscope, this is a part of the brain known as the hippocampus, which is involved in memory and learning and is particularly at risk in prion diseases. And this is a layer of cells here, this purple color called the pyramidal cell layer. If you look at that, you see at 8 weeks of age, you see these holes appearing in the brain, the spongiform changes. And by 12 weeks of age, more holes, and now this cell layer has completely died. In fact, the whole hippocampus has shrunken. But now if you look at the double transgenic mice, at 8 weeks of age, they're still making prion protein, so these pictures are identical to those pictures. But now at 12 weeks of age, when these cells would normally be dying, you can see they're not, they've all survived, and remarkably, the spongiform changes have actually gone away, so there's actually been some recovery of the pathology. So we can target the prion protein in an adult brain without major consequence. That causes a complete block of development of the clinical disease in mice with an established brain infection, and the early changes appear to be reversible. So the general principle of what we're trying to do here, we know for reasons I'm not going to go into this morning, that for the normal prion protein, which is shown again here, to end up in these long fibers that I've shown you, we know that it has to largely unfold to do it. It has to almost completely unfold into that string of beads again in order to end up in that form. So something that binds to it and holds it into its normal form will prevent that process happening. And as I mentioned already, prions are not a set of identical things in the brain. They have other components to them that are slightly different. And so if you develop drugs which just bind to the disease-associated form, the rogue forms, there's a danger that you'll get the rapid development of resistance, and that there'll be some minor component to which your drug doesn't work so efficiently that will then take over. But if you bind something to the normal form of the protein, and this is an antibody is roughly a Y-shaped molecule, hence that symbol, then it doesn't really matter what strain is here, it doesn't matter what mixture is, because they all require the normal prion protein in order to propagate. If there's no normal prion protein, it's all gone. The prions can't propagate anymore. And the brain turns out to be relatively good at getting rid of prions. That might seem surprising, but a brain that can't propagate prions gets rid of them quite quickly, in the order of about 10 days. So if we stop prions propagating, then we hope that the normal brain's clearance mechanisms will do the rest and lead to a cure of the infection. So development of antibodies. So the idea is, as I've mentioned, these antibodies bind to the normal prion protein. They hold it away from the prions and prevent the prions propagating and getting a hold. Now, as I've mentioned already, we don't make our own antibodies against prion protein because it's one of our own proteins. So what we did is take mice in which the prion gene has been knocked out since the earliest stages of their development. So these mice have never seen the prion protein before. We now inject them with human prion protein that we've made by genetically engineering that into bacteria. We can make the human protein in large quantities, inject that into these mice. Now, these mice have never seen this protein before. They think it's a foreign protein, and so they make antibodies against it. And we made about 100 different antibodies that way. And then you can make large quantities of these antibodies and inject them into mice that are infected and see whether it stops the disease or not. And that work we published quite a long time ago, about 15 years ago now, actually. We showed that these monoclonal antibodies can block prion propagation in mice and delay the development of the disease. And again, I'm showing you some survival curves here. So you start off with 100% of the animals alive. You inject them. RML is a particular strain of prions we use commonly in the laboratory. And you see after a period of time, they all die. These are mice that are injected with normal brain rather than infected brain. And this is the normal lifespan of animals in the laboratory. So laboratory mice live about 2 years if you look after them well. And so you see this is natural. The mice are dying of natural causes here. They'll all be dead by around 700 days. Now, if you give them one of these antibodies we've been studying, called ICSM-18, as you give the mice increasing concentrations of this antibody, you start seeing increasing durations and increasing survival. And in fact, at the optimum dose of 2 milligrams per week, about half of the mice survive to old age. And in fact, when we then autopsy these mice and look at their brains, we can't find any prions in them. So they're apparently cured of the prion infection. So this is very promising. These are mice that were injected by the peripheral route, not the central route. It's more challenging to once the disease is established in the brain, to actually get sufficient antibody into the brain. And I'll come back and talk about that in a minute. At higher doses, over long periods of time, we have seen some inflammatory side effects in these animals. That's not surprising. Treatment of a CNS disease with antibodies is often associated with some sort of side effects. Our experience is this is, however, reversible if you stop or reduce treatment at that point in time. Whether these side effects will be a problem for humans, we don't know until we try that. But as I'll come on to say, we've engineered the antibody to reduce the possibility of unwanted immunological side effects. So we've achieved a so-called proof of principle. In principle, this approach should work. We ought to be able to treat infected individuals before the prions get into the brains. That applies to individuals who've been accidentally exposed to prions, for example, during medical procedures. In principle, we could give them an intravenous shot of this antibody and prevent them ever getting the disease. Of course, what we really also want to do is to see if we can treat patients with the established disease. And that's going to be a question of, can we get enough of this stuff into the brain? To begin with, as I'll come on to talk about, we're going to give this by intravenous injection and see if we can get sufficient driven into the brain. If not, we may have to give it directly into the spinal fluid. So we were funded to do this around in 2007 by the Medical Research Council. Now, you can't treat patients with mouse antibodies because, again, your immune system would recognize this as being a foreign protein and would destroy it. But the technologies existed for some years to make so-called humanized antibodies, and by genetic engineering methods, you can turn a mouse antibody into a human antibody, and if you do it well enough, you keep its properties, but it still becomes human. And of the 100 antibodies we studied, 2 of them were particularly promising. We made human versions of both of them. And we made 2 different forms. Again, the technical details don't matter, but there are different so-called isotypes of antibodies. And we focused on what's called the G4 isotype. And the reason is that has much less unwanted immunological effects, because antibodies do a whole range of things. They don't just stick to something. But all we want is for this antibody to stick to the prion protein. We don't want it to do anything else. But what antibodies have evolved to do is that the other end of the antibody that's not sticking to your target recruits other parts of the immune system, particularly to bring in other cells and other reagents that will kill cells. We don't want this to kill cells. We want to stop killing cells. So we've chosen this particular type that isn't very good at interacting with the rest of the immune system, and we hopefully will see less chance of side effects. So this humanized G4 version of ICSM-18, which is rather a mouthful, so we call it PRN100, has now been manufactured for clinical use. And it's been through a whole series of safety testings that are required from a regulatory point of view, a key one of which is a single ascending dose study in primates, in macaque monkeys, were treated with doses up to 200 milligrams per kilogram. That's a huge dose, I should say, intravenously, much higher than we'd planned to treat patients with. And I'm pleased to say there were really not any significant side effects to worry about there. And the antibody, the human antibody, works essentially just as well as the mouse antibody did. So we've retained its properties while converting it into a human form. That's a vial of the stuff. Now, actually, before I do that, show you this video, the son of one of my patients, who's an expert in animation, has kindly made a video showing how the antibodies thought to work. And the daughter of another of my patients, who's an actress, Liz Bichard, has narrated this. So I'll show you this as a summary of what we essentially think is going on here. I hope the sound's going to work. Who was struggling with that a bit earlier.
Prion diseases are fatal degenerative conditions which progressively affect the function of the brain. Our brains each contain 100 billion nerve cells, or neurons, which are connected by branches of the cells called dendrites. These connections allow electrical signals to travel between the nerve cells, and this communication between the cells is key to how our brains work. There are many different protein molecules that normally sit on the surface of the nerve cells, and one of these is called the prion protein. Thousands of these sit in clusters all over the cell surface and play a role in the normal function of brain cells. Prions, the agents that cause CJD, are formed from misshapen or rogue prion proteins that stick together to form chains. These chains grow by attaching more and more normal prion proteins, which also become misshapen. The growing rogue prion protein chains tend to break up as they grow, thereby producing more chains. These in turn grow and break, creating a chain reaction. The multiplication of prion chains damages the connections between brain cells, and ultimately the cells themselves begin to die. As this progresses, the normal functions of the brain begin to be lost, causing problems with memory, thinking, speech, balance, and movement, ultimately leading to the death of the patient. The human body normally produces molecules called antibodies to fight infections from germs that invade the body. However, since prions are formed from one of the body's own normal proteins, in the case of CJD, our immune system does not do this. We have therefore artificially created a human antibody called PRN100 that binds tightly to the normal prion protein. This can be manufactured in large quantities for use as a drug given by injection. The aim is for the antibody to bind to the normal prion proteins on the cell's surface and block their interaction with the rogue prions. This prevents the prions growing further and stops the chain reaction. We know that the brain can slowly get rid of the remaining prion chains by its own natural mechanisms once they have been prevented from growing.
So I hope that gets across the general concepts of what we're trying to achieve here. Now, having made this drug, we've had this drug now for several years, I'm increasingly embarrassed to say. We anticipated, and this was our original timelines for conducting a formal clinical trial with this drug, first in the UK, and then we hoped with international colleagues to widen that. But we were unable to get sufficient funding. Doing formal clinical trials, which are highly regulated affairs, are extremely expensive. They cost millions of dollars to do, and we weren't able to reach that level of support for what is, of course, a rare disease. And these were the things that we thought we were doing. So we were unable to progress this, although we had been in discussion with the UK regulator and had very positive encouragement from them. So in the absence of money to do a full-scale clinical trial, what we've proposed to do is to offer the drug, which, as I say, has been around now for some considerable time, to a number of patients with sporadic CJD, with a rapidly progressive form of the disease. As I'm sure you know, the average life expectancy in patients with sporadic CJD is about 4 months. The average life expectancy from the point of diagnosis is about 6 weeks. So it's rather similar ethically to the situation with advanced terminal cancer. And we think there's a strong case to simply offer this drug to those patients on a compassionate use basis. And that's what we've been trying to achieve for some time. We plan to give the drug in the first instance intravenously at increasing doses over 48 to 72-hour intervals, obviously starting at very low dose in case we have unanticipated side effects. If we don't achieve sufficient concentrations in the therapeutic range in the brain with the highest doses, then we'll shift to giving the drug directly into the fluid-filled spaces of the brain, which is a straightforward, it involves a neurosurgical procedure, but it's a very straightforward procedure. And that will ensure that we achieve adequate concentrations in the spinal fluid. So where are we with this? As I've told you, we've been at this since 2007, 10 years now. We've done all the production, safety testing was completed. We didn't get sufficient funding for a formal clinical trial, nor were we able to attract a commercial partner, despite extensive work over several years. So we proposed about 18 months ago going forward on a compassionate use basis, and the hospital I work at, University College Hospital, established an oversight committee to help us with that. And the patients, including the patients that made that wonderful video, set up the Cure-CJD campaign in the UK to raise funds to do this compassionate use, and they'd successfully raised all that money about 6 months ago. Unfortunately, we're still waiting for the legal agreements between the multiple parties that are involved in this, including the MRC and the hospital and University College, to actually for the hospital to actually be happy it's in a position to give this drug. So I was hoping that I'd have something more positive to say when I arrived here in July. When I was kindly invited to give this, I thought, well, we should have treated the first patients by that stage, but alas, we haven't. But I'm hoping we're going to be there before too much longer. It has been incredibly frustrating. These are some of the committee from the Cure-CJD campaign that raised the money to do this. These are the members of the National Prion clinic staff, some of whom you'll know are nurses and doctors that see patients all over the UK with these diseases. And the work on immunotherapeutics, the laboratory work's largely been done by Azi Khalili, the lead scientist on this, with a lot of help from Caroline, Simon, and Nicola, and many others. Simon leads the clinical research in the unit. Many of you will know Simon, I'm sure. Sorry, I'll finish there and happy to take any questions.
So if I may, this is amazingly complex and challenging project. And I think that there are similar examples of those therapeutic intervention with some encouraging results in Alzheimer's disease. So why do you think it's so difficult to get ahead with therapy of invariable, fatal, rapidly progressive disease compared to the Alzheimer's disease, which is now routinely approached with similar strategies and tested in many different clinical trials?
Yes, a number of trials have been done with therapeutic antibodies in Alzheimer's disease. None of them have succeeded, but a lot of work's been done. But Alzheimer's disease is obviously a very common disease, and there's a huge commercial interest in the pharmaceutical industry to do that, huge interest. And so they've been willing to fund trials that have cost hundreds of millions of dollars to do. But CJD, as we all know, is thankfully a rare disease. It's one of enormous interest to all of us in this room. But for commercial partners, it's not so attractive an idea. And I think you have to recall that when my unit was set up and the work we did on the antibody there cost £8 million. It's a lot of money for an academic group. But at the time we were funded to do that, there was real concern in the UK that we might see an epidemic of variant CJD. There was particular concern that it also might be transmitted by blood transfusion, as it seems to be have been able to do. I think now it's clear that there won't be many more cases of variant CJD in the UK. There isn't the impetus anymore to spend large sums of money on this, and so we have to find more imaginative ways of doing this. So I think the compassionate use is the way ahead, in that it's much less expensive. It still takes a lot of money. It's still costing about £450,000 to do that. But it's been possible to raise that money with enormous support from our patient community in the UK. So I think we have to hope that if we have those data from that compassionate use and they're promising, it may then be possible to persuade a pharmaceutical partner to get more interested. At the moment, this is too high risk for them. Pharmaceutical companies are pretty risk averse. They want to see preliminary evidence that the drug can get into the brain, that there are no particular safety concerns. I think at that stage, even in a rare disease, they might be interested. But at the moment, it's not an attractive prospect for them. So it's still in the hands of academics like me to try and move it forward with what resources we can manage to muster.
A second very short question. One proposal by Street Matter is that PRPC may be a receptor for misfolded amyloid beta. So your antibodies could be, in theory, blocking the toxic effects of amyloid beta in the Alzheimer's disease. Have you done any experiments to test it or test this idea? Because that would clearly broaden the applications of this approach and this specific antibody.
Yes, we have. That's a very good question. We have looked at that, and we published some work a year or two ago looking at a rat model of Alzheimer's disease, where we injected soluble extracts of A-beta from brain of individuals who died of Alzheimer's disease into rats. That caused defects in the rat brain, which could be corrected by giving PRN100 intravenously. So there is some, albeit preliminary, animal evidence that this might be relevant to Alzheimer's disease too. You might think that means there'd be a lot more interest in developing the drug for CJD in the first instance for that, and believe me, I have used that argument quite a lot to try and get the pharmaceutical industry more interested, because obviously there's a definite interest in developing drugs for Alzheimer's disease. There isn't really for CJD. But so far that hasn't enabled them to sort of take the bait, as it were, and help us get further forward with the CJD treatment. But yes, it's possible in the future, if the drug turns out to be safe and sufficient gets into the brain and we have promising results, it may be worth in the future doing some human studies with Alzheimer's disease too.
What type of liability issues do you have with the facilities that you administer the drug? Do you have liability issues?
Yes, there are all sorts of. I mean, this is sort of beyond my pay grade. I just have to sit and watch as the lawyers from the multiple institutions discuss all these things. It's rather frustrating. I feel it ought to be exposed more to the patient community, who are probably not so worried about liabilities when you're facing a 6-week life expectancy and no alternative treatments. But yes, the institutions do worry about the legal consequences of doing this, that, or the other, and this is where we are at the moment, with them seeking all sorts of legal undertakings from each other in terms of before the hospital's happy to go ahead and do this. I think this is a problem for rare disease. It's not just about CJD, but there are a number of rare diseases where specialist treatments have arisen, and where it's a disease with no alternative treatment, certain death. I think we do need a different model, actually, probably more patient-driven, that, you know, let's stop all these lawyers arguing for years about this, let's get on with it. But of course, that's not in my hands.
If I could mention something here. Thank you very much for this presentation. I find it fascinating. I also find it appalling that there is a drug out there that none of us knew about, really, and that the legalities of this happen to be preventing this drug from happening. So my question is, we can talk about that all day, but how do we as individuals help on going forward with this? I understand you're doing a lot in the UK. What can we do in the United States to try to get this treatment in progress for us? Because this disease, as we know, kills. So why can't we just go ahead with compassionate use? But the question is, what can we do as individuals?
Well, thank you. As I showed you, there is the Cure CJD campaign was formed in the UK to produce the funding for this. And they're, needless to say, concerned about this too, to put it rather diplomatically. So they've been speaking to a number of individuals in all these organizations to try and move things on. And I'm sure they'd welcome hearing from you. I think some international support might be extremely helpful. And you can see their website with all the contact details. It's curecjd.org, which also has that video on it. If people want to watch that again, that video is on the website there at curecjd.org. I'd encourage you to get in touch with the patient group there, who are very active in putting pressure on to try and clear this logjam at the moment.
So further on that excellent question, we've got about 200 countries in the world. It sounds like this logjam is going on in England, right? Is there anything that any other country can do? Or is there a way of you partnering with some country where the laws are...
Well, you're not on that, so you need to be short-worded.
So if I may repeat the question, is there any way to partner with international organization or other countries or organization in other countries to facilitate or accelerate the clinical trials?
Yes, if it were up to me, and I'd pack the drug up and go to wherever it was possible to offer it to our patients. But it's not up to me. The drug's owned by the Medical Research Council, and it has to go through a process before it can be released. Those processes, of course, are there to a significant extent. There's more to it than that, but some of the processes that are holding up are there, of course, to protect patients in terms of, one has to be happy that the drug's been manufactured to the right quality and various boxes are ticked, and I fully understand that, but that needn't take very long, particularly as the drug was ready for a full-scale clinical trial anyway, so most of the regulatory things had already been done. But yes, in principle, it could be done anywhere there are patients with CJD in an appropriate medical facility. But of course, other facilities will also have their own institutional hurdles that are there before they would be happy for a drug to be administered in their hospital. So it's not necessarily easier anywhere else, but I'm certainly open to suggestions.
Congratulations on your work. Very exciting. Also, I can imagine very frustrating with 10 years of searching for commercial partnership and funding. Similar comment that's been brought up. in the United States, in that same 10-year time frame, there's been lots of exciting progress through the orphan drug incentives in the United States, and micro-orphan conditions like this, where it literally affects hundreds, not thousands or tens of thousands. Have you pursued some of the incentives in the United States around the orphan drug program, which has been successful? Companies are making money, patients are getting treated, and the ecosystem is working finally. Unfortunately, not for this disease, but for lots of other rare diseases. Have you looked into all of that?
We have looked at that. I mean, it does lower the threshold a bit for companies in that it makes it easier for them to get a drug approved, and they have a longer, period of protection and so on to market the drug. So there are some advantages in that, but we unfortunately we haven't got to that stage yet. But I think if we can do the initial studies, we can ultimately we have to have a pharmaceutical company involved. I run an academic group. We can't market a drug. So we have to have a pharmaceutical partner. And I think those sort of things make it a bit easier for a pharmaceutical partner to see how they've got a business case to do this, and their investors, will approve it. But we haven't even got over the first hurdle yet of doing what's called the first time in human study. And people want to see those sort of data before they'll seriously engage.
Thank you. Thank you for your talk. My question is, you mentioned using this drug in sporadic CJD after onset of symptoms and diagnosis. Can you talk about the potential use for hereditary forms of the disease in asymptomatic or pre-symptomatic patients who are mutation positive?
Yes, a very important question. The reason we're looking at sporadic CJD first, and indeed the more aggressive end of sporadic CJD, is essentially for ethical reasons, because the clinical situation is hopeless, and death is inevitable in weeks usually from diagnosis. And so I think the risk-benefit situation is very obvious. When you're putting a new drug, particularly a so-called biologic drug like this, the antibodies are very powerful molecules. There is a risk of doing significant harm, and therefore that particular group of patients may be willing to take that risk. I know I would in that situation. So, that's why we're looking at that patient group first. And we will, of course, assuming we get to do this, we'll have safety data from that. We'll know whether there are any problems in the short term or in the longer term giving the drug. Now, if it turns out that the drug's safe, and as I say, the safety testing we've done so far in primates has been encouraging. If we find that it's actually quite a safe drug to give intravenously, then absolutely we'd then want to go on to look at other groups of patients. And there's for the reasons I explained, because we're taking out the normal protein itself, we're basically switching off the carburetor. The engine can't get any petrol anymore. So that works on any model of car. So this should work against any strain of prawns. They all have to have prawn protein in order to replicate themselves, and it should work in the inherited forms too. And of course, a particular advantage in the inherited forms, as you allude to, is that because we can do a genetic test in those individuals, we know who's going to get the disease. And so in principle, if the drug is safe enough, if the drug carried significant side effects, that might be okay. If you've got the disease, you might put up with that. If you're going to treat healthy people to stop them ever getting the disease, then you want to be sure the drug's very safe. So it will depend on how safe it is. But if it turns out to be very safe, the most optimistic view, then we'd want to treat patients carrying mutations and prevent them ever getting the disease. That would be the aim.
Thank you. I'll speak really loud.
I'm told this has to be the last question. Sorry.
Thanks for your presentation. I really appreciate you coming here and sharing. I believe you've done work in the past on RNA interference. You didn't talk about it today, but can you speak as to whether or not you see that as a therapeutic option in the future?
We did the graduate student who did the work I showed you in transgenic mice did some work with RNA interference as a potential therapeutic. I mean, there are a number of companies looking at that, and I think perhaps more promisingly using antisense oligonucleotides to target expression of proteins. And I think there is some progress in that area. Conceivably, it could be used. Again, you've got the issue of how to get sufficient into the brain and throughout the whole brain to actually have a significant effect. And so that's also going to, I think, be a very challenging approach. I think at the moment I would favor the antibody approach. Of course, we've got limited bandwidth. Doing this has taken all the effort we've got, to put it mildly. So what we don't want to do is to start thinking, oh, well, that's promising as well. You've got to focus on what you're doing. And as you can see, it's taken us 10 years to get to this point. So, yeah, I mean, there's potential from a number of other areas of looking at how you target gene expression. But I think that's for others to pursue.
Thank you.
Doctor, can I make one comment? And first of all, thank you for coming.
Do you have? Right.
It's kind of relevant, though. Something that the doctor said. I'm actually a pharmaceutical products liability attorney. I sue drug companies for a living. And my mom was diagnosed with CJD in October of last year, and we were at UCLA, and I was begging them for anything experimental. I mean, there is, and I can tell you from a legal perspective, when someone is dying, you know, both ethically and legally, you risk whatever if there's even a slight chance of trying to save them. And, you know, to the point where I was even willing to fly to UCSF, and I found out their study on quinacerin had been suspended or failed. But, you know, we're about to go visit our congressman on Monday, you know, to get funds. But if it's a matter of simply changing the law, so for a disease like this where you know death is imminent, and you have a medication sitting on your shelf that could potentially help, I mean, it's a no-brainer. If the law needs to be changed or to make an exception for that, I'm happy. I've lobbied Congress in the past on behalf of injured consumers against the pharmaceutical company. I'm happy to do that because it is just obscene. Had I heard your talk 6 months ago, I literally would have broken into your lab and stolen whatever vials you had and brought them. And you know, it is obscene. I mean, there is no risk of liability or anything when the person is on their deathbed, literally, and you can only help. And it is, I mean, you gave one of the most fascinating speeches I've ever heard, and I've gone to, and my mom was an infectious disease doctor herself, so she carried me around to medical conferences all the time. I mean, yours was one of the most fascinating I've heard, but also, I mean, you have no idea how heartbroken I am. And I'm sure others in the room listening to what you just said, because it is, so whatever assistance you need, I mean, I don't know how the UK system works, but in the US, I am happy, and I'm sure others will be happy, you know, because maybe it may be hard to get money from Congress, but it's not difficult to get them maybe to make an exception to rules or alter the rules, whether at the FDA level or on a state or federal level.
Well,
thank you.
Firstly, I'm very sorry to hear about your mother. But secondly, all I can say is I utterly agree with you. I just wish I could do something. It's not about money now. We have the money. It's about getting these obstacles out of the way, many of which I think are just misplaced. And it's fairly obvious to the patient community this is misplaced and this should be progressing. As I said to a previous questioner, I can give you the details of the patient advocacy group in the UK, and I'm sure they'd be delighted to hear from you and have your help too. I can also, if you catch me after the talk, I can give you my card and perhaps we could get in touch and see how you can help. I'd be very grateful for that. Thank you.